And so the panel discussion is titled Research Ideas to Generate Evidence for Action. So it's really application of these novel technologies, these genome editing technologies now in terms of application for the future. And so if we could have our first presenter back, Dana, if you could give us your one slide presentation. We also have Mark Hahn and Rebecca Frye that will be presenting, but we'll start with Dana.
All right, good morning. It's good to be back, and the same caveat applies today. I'm still coming at this from a little bit different perspective than the majority of the conversation. But I thought that it might be nice for us to consider the similarities and the differences between genome editing and epigenome editing. And I wanted to break this down on sort of 3 different levels, starting with these laboratory approaches that you see up here, and then thinking about how this may be applied to clinical and even population environmental health practices. So one of the things that I discussed yesterday with the trial of piRNA as an epigenome editor that will induce DNA methylation is the fact that this is a targeted method, and many of the current epigenome editing tools are what we call global editors. So they demethylate or methylate the entire genome. There's also editors that act on the histone code. Certain labs, including the laboratory of Rudy Yanish at MIT and as well as Gerbosch lab at Duke, have also begun to use CRISPR as an epigenome editor. But there's various efficiencies and sort of how long these epigenetic marks last through subsequent cell divisions. But if we are successful in adopting some of these techniques, some of the tools in the laboratory that we might use are the ability to knock in or knock out epigenetic mechanisms, sort of similar to what we're talking about here today. And one example that I thought correlates well with what we've been talking about with genome editing, and when you discover through GWAS or other methods a list of genes that are changed in response to the environment that lead to a phenotype, we also are doing a lot of this with epigenetic epidemiology. So we have arrays now that can look up almost 1 million different CPG sites at a time. And what we've seen in environmental health, and there's a review that came out a few months ago in Environmental Health Perspectives that was put forth by the EPA and NIEHS Children's Environmental Health Center researchers. What you see in environmental health is that the effect sizes of the environmental agents on the epigenome are quite small, especially in comparison to things like cancer and other disease phenotypes. But they're also quite meaningful. And so once we do some of these million SNP or million CPG site arrays, we might be able to then move our top hits into an epigenome editing cell-based approach to begin to figure out which ones are the top priorities. So there are some great parallels there. The other thing I'd like to talk about is there are some really interesting potential applications for epigenome editing in the clinical setting, including co-administering epigenome editors with these genome-wide pharmacology agents, so cancer chemotherapies that erase the entire methylome. If we were able to also co-administer some agents like piRNA to specific regions of the genome we know that we do not want to be demethylated, this could also be a very interesting approach. And in our conversations yesterday and today, especially coming after Dr. Brayboy's presentation, there are potentially some application of this for during IVF and egg cryopreservation if we can identify regions of the epigenome that are changing with these alkylating agents and such not. And then finally, I know we're not necessarily talking about human applications here or ethics, but one could imagine that epigenome editing may have a lower bar to genome editing when thinking about how we apply this to either acute or long-lasting environmental exposures. And we may, the same way we think about using nutritional approaches to counteract some epigenome changes, there may be some use in the future of using epigenome editing to protect ourselves against environmental exposures. So thank you for your time.
So I think we'll hold questions and comments till after we hear from each of the three panelists. So the next person we're going to have presenting is Rebecca Frye. And Rebecca, if you could come and give us your one slide.
So yesterday I had been discussing translation. How can we translate from cell culture to human populations? And in this, as I was thinking about how we might apply this, I really focused in on regulatory aspects of chemical science and risk. So some of our gaps are how do we set limits for chemicals in the environment? And one of the struggles that we face generally is how do we set limits and understand those limits in the context of vulnerable populations. So my thoughts were that we could really be using gene editing to help us to understand variants, human population-based variants that influence susceptibility. So if we understand susceptible populations, that will influence the levels at which we're regulating chemicals. So for example, if we can use CRISPR-Cas9 to genetically engineer cells with different, that are knocked out for different genes, and we screen those, and as we were talking about yesterday, right now we're screening for cell survival, but we should enhance our suite of phenotypes that we're thinking about. We establish then this, we can establish sensitive and resistant populations based on the genes that have been targeted. So then once we've identified these genes, we can begin to understand the polymorphic variants that are associated with vulnerability to environmental toxicants. We can use knock-ins to understand which of these variants are really driving susceptibility. And then we can translate that into human populations, looking in human populations for those same variants. So that's just one way that I'm envisioning the use of gene editing for regulatory decisions. So.
Thank you very much, Rebecca. Mark Hahn will be our last presenter here before we start the discussion.
Okay, thank you. So I really kind of struggled with this charge, and I struggled in part because, and here's the disclaimer, I know nothing about this topic of decision making, so I will say that up front. And I thought about what are the what are the guiding principles that I would follow in trying to come up with research that would inform decision making. And one of them is that decisions should be based on understanding. And maybe that's self-evident, but I think it's good to repeat it. And therefore anything that enhances our mechanistic understanding will or should contribute to better decisions. Okay, I think we can all agree on that. A second one is that improved understanding of mechanisms underlying species differences should improve the process of extrapolation from model systems to humans. And so I think that's one area where improved mechanistic understanding enabled by genome editing could make a contribution to, let's say, risk assessment, for example. And then the third thing is not a principle. I thought that maybe these two panels we're doing this backwards, and that I would actually like to hear from the decision makers first, telling me what is the information, what is the critical information that they need to overcome obstacles that they see in their process of making decisions. And so I'm kind of very much looking forward to hearing what the next panel has to say, and then maybe we can come back to you with some more ideas about how we can help. So with that said, I did take a stab at this, and I addressed two areas. The first one is the business decisions, and something that we haven't really talked about over the past 2 days, which is a drug development pipeline. Note the disclaimer again, I know nothing about this topic. And I think I think it's already underway with pharmaceutical companies, and that is using CRISPR to develop new screens for drug targets, for validation of targets, and for developing models of human disease, of modifiers of human disease, and of adverse events. And some of these are humanized models where they're taking model systems and knocking in human genes or SNPs to try to recreate in an in vivo or a cell model something that occurs in humans. The second one I took a stab at was the regulatory question and the decisions about how to set exposure limits. And what I thought is that setting exposure limits used to be easy when all we were concerned about is preventing people from dying from a chemical exposure. Okay, I think it was easy. I wasn't involved in that, but it certainly was easier. And now we're concerned about more subtle effects that are occurring, and these generally occur at lower levels of exposure. And so it's a hard problem. And adding to the difficulty of the problem is that exposure and effects now are often separated widely in time. And so we're trying to link an early low-level exposure to some effect that's occurring later on in the lifespan. And so this is just following up on something that I addressed yesterday, that again, we need to better understand mechanisms underlying the developmental origins of adult human disease susceptibility. And I'm going to break the rules and show a second slide, which is just a repeat of the slide I showed yesterday at the end of my talk. But I've filled in some of the references for what I think are some of the most exciting developments that will help us to study these mechanisms of developmental origins. And that is some of the new techniques for lineage tracing and biological recording. And we heard a really elegant presentation this morning providing an example of one of several possible mechanisms for biological recording of events that has been published recently. So I will end there.
Okay, Mark, thank you for that presentation. So I guess I'll take the moderator's prerogative, and maybe I'll kind of start this off and see where it goes. And Mark, I want to build kind of off of not your second slide, which you weren't allowed to do. No, I'm just kidding. But the first one, and it was the example you gave about drug development, and that's a perfect example of when we know what the target is, we can use these gene editing techniques to really build on that. But when I'm thinking about environmental health sciences and some of the challenges that we have, and Keith touched on a lot of these in his presentation, we don't often know what the target or targets are. And so how do we begin to utilize these tools from some of the very nice mechanistic, elegant mechanistic studies we saw yesterday, where people really knew what the target was. They were clearly after a specific target, and try to begin to utilize these techniques more in a broader kind of hazard identification approach, which Keith, is kind of where your work and your group's work at EPA is headed to. And I'm just wondering if maybe we could start with that, and we'll just see where the discussion goes. Sorry to put you on the spot.
Well, I mean, I think, is that on? I think I covered a lot of that. We went from an initial model where we used a lot of in vitro assays that had specific targets designed into them, and then have broadened our coverage by going to a transcriptomics approach. And as I said, we learned that in these types of approaches, you find that environmental chemicals have lots of targets, so which really are the significant ones. We partly address that by trying to model the in vitro results using animal data. So a lot of the chemicals we screened, especially early on in ToxCast, were ones where we had extensive guideline studies, a lot of pesticides, for instance. And so we would see how those chemicals behaved in our in vitro assays and use those data then to try and predict the animal toxicity. And we were modestly successful, at least. But again, I mean, there's a couple of problems with that. One, we're predicting animal toxicity, and we're really interested primarily in human toxicity.
Yeah.
And so we would like to go beyond that, and that's where some of these approaches, like I described, where the functional genomics approach may give us clues about what really are the critical targets for specific chemicals, might be helpful. But we're in the very early stages of that, and it's going to be hard to judge how universally successful that may be.
Yeah. So are you envisioning maybe in the future creating libraries of cells that have different pathway components that have been eliminated? And then that kind of gets at the question of, well, a lot of those kinases, phosphatases are absolutely required for these cells and their biological activity. And if you eliminate them, then can, you know, first of all, will they survive? Will they function? And is that going to be a feasible kind of strategy for hazard identification?
Yeah, I mean, I think what we would like to be able to do is, again, use a tier 1 screen that's a global type of screen. Transcriptomics is the most obvious choice, and that would allow us then to narrow down where we would focus our efforts in characterizing that chemical. And it would be, there is probably going to, well, there are a defined number of toxicity pathways. We don't know what that number is right now, but, you know, it is a defined number, and you could eventually have models that have assays already set up designed to look for those specific effects on those specific pathways to confirm what you've seen in the tier 1 screen. And like some of those, like the issue you said with a critical kinase, for instance, I mean, those are all technical challenges that are very approachable. I mean, if you knock down the expression by 50%, the cell may still survive, but it's now going to have increased sensitivity to inhibitors of it. And these kinds of genome editing tools allow you to do that. So if we know what the targets are we want to cover, the technology is there to build the assays where you can quantitate the effects of these chemicals on those specific pathways, but we're not there yet in understanding what the targets are that we would need to cover for such a broad area of environmental toxicology.
Lisa Aylward, Summit Toxicology, and I think you sort of just answered the question I was going to pose, which was, are these tools, you know, so my impression in following the ToxCast and Tox 21 programs is that, you know, we're sort of looking under a lamppost still with the assays that we use so far. That is, we've used assays that were already available. We've used, you know, genes that we knew were a priori interest. And so the question of what biological response spaces that we don't cover well with those assays, you know, more complex, you know, responses where we, you know, have to essentially recapitulate an organ or a more complex biological system. And the question is, you know, does the ability to more easily manipulate a variety of biological systems. So we've talked about the fact that, you know, that these technologies allow us to actually to be able to manipulate all kinds of different species at various levels of development and organization. Do these tools give us a way to more intelligently or supplement or design assays that help us broaden our space in the high throughput context? And is there, you know, are there And I think, I think what you said is that's kind of, we can envision that and we can do that, but I wonder if there's any, specific development going on in that arena.
Yeah, I'm not aware of any applications along the lines of what you suggested. I think people think about them and envision them, but I'm not aware of anybody actively doing that right now.
Yeah, in terms of better lampposts to be using these technologies for Gary Ginsberg, State of Connecticut. From a public health perspective, we also want to be focusing on what conditions are we concerned about in the population and what explanations, biological explanations, do we have for those conditions that can be assisted by these gene editing tools, so that as we learn what is causing disease, better understand what's causing disease, what pathways, what epigenetic modifications, what SNPs are associated with things like what I brought up yesterday, the increasing rate of liver cancer in the population related to metabolic disorder. Maybe then we'll have some idea of the changes from a background disease perspective that these chemicals at very low dose, which is where they may have their major action in a biological system, rather than at overloading systems at high dose, but at low dose, what they may be adding to. And so if we bring in the perspective of what we're learning with these tools from medicine, pathology, into our toxicology method, maybe that will help us focus on which genes, and a good example is what we're starting to learn about breast cancer and a highly penetrant gene like BRCA1, and seeing that the epigenetic modifications of BRCA1, it's not just an all or nothing. Either you're a knockout or you have it or you don't, and then you have to worry about a genetic susceptibility, but there may be environmental modifications of the promoter of BRCA1, as we're starting to see show up in the literature, that do the chemicals we're concerned about, or environmental chemicals, do they modify that promoter? Can we start developing an array of highly penetrant genes that affect human risk for diseases that would then be important to probe in vitro, whether it's in Tox21 or it's through gene editing methodology? So I would just, I was part of the Science and Decisions, where we tried to talk about interaction between toxicology, the intersection between toxicology and background disease processes, and bringing more of that perspective into the advancing research would be great, so that we have people at conferences like this that are not only working on the AOPs from a toxicology perspective, but also the AOPs from a, what's the systems biology disease process that we should be focusing on that's prevalent in the human population.
I would just respond that I agree wholeheartedly. I think that's a great comment and something we all need to keep in mind.
I also have a quick comment. One of the things that we've struggled with in incorporating epigenetics into risk assessment is sort of what is adverse. And when you do an epigenetic screen, you see all sorts of things, some adverse, some neutral, some adaptive. And I think we also struggle from the field being more descriptive and less mechanistic. So I do see that some of these genome editing tools could get us to a better place where we might be able to actually hone in on which of the data in an initial sort of epigenome-wide screen is what we should focus on from a regulatory standpoint.
Melissa Perry, George Washington University. I raised this in part yesterday, but I'll raise it anew in the context of the question about advancing research and having the most impact on consumers, business policy, and risk assessment and regulation. And that relates to the data and the methods in which we're looking at associations and being very facile and being multivariable or multi-factor. And I think this question goes to Keith in particular. To what extent are gene editing data presenting new challenges and new opportunities in being able to evaluate, test, and verify multifactorial relationships. As an epidemiologist, I often lament that our methods are not robust and powerful enough to look at things like mixtures and synergies. And so my question is, how can we push forward with these new data, this new method, these new mechanics, in order to get at the heart of relationships as it relates to mixtures and synergies and interactions?
That's a really good question. Specifically with respect to using these tools, I think you could sit down and come up, it probably would work better from sort of a case study perspective, where you could set up some potential models where you expected to see synergistic effects and where you know some of the key players in that. And then these type of genome editing tools could be used to devise experimental systems that would validate those ideas. And then assuming that you are able to validate those ideas, that same type of a system could be modified into something that you could use as a more universal test system to look for those types of synergies for unknown chemicals.
Mm-hmm. And I assume that all of the members of the panel in some way, shape, or form work with biostatisticians or statisticians and quantitative methodologists. And to what extent are they saying to you, oh, these are new interesting data, or not? To what extent are we pushing a new boundary that could invite new capabilities in terms of modeling and data analysis?
So I'm the newbie, and I'm just now starting to talk to biostatisticians, and the concept that MDRs or multi-drug resistance transporters are not pathologic, that they're actually protective, is sort of throwing people for a loop. And then looking at them in normal tissue is a challenge too. And so I think that I'll have more information in the next year, because now I have these large data sets that I need help analyzing. And again, I think, as you probably know, reproduction is still not as studied as it maybe should be. And so really trying to have that conversation about how the ovary works is another big challenge. And so I haven't gotten into gene editing yet, but I think that just as a neophyte, I think in science, that it's a challenge to try to communicate some of the issues that I have. But as I try to make the knock-ins or the SNPs that I want to do, I think that that's going to be very, very interesting and an interesting conversation. But also, I wanted to just comment on one thing that you talked about for BRCA. So we see a lot of patients who present first with infertility, who have BRCA, and they may have infertility decades before they actually develop their ovarian cancer or their breast cancer. And so I think it's really important that we understand that BRCA does more than just gene editing, that it does so much more. It probably is protective to the ovary in some way, and maybe the vulnerability is to toxicants. And so I just wanted to bring that up as well. Thank you for your comment.
John Balbus with NIEHS. I just wanted to pick up on the comment that when we're looking at these applications for drugs, the drugs are targeted, whereas with the environmental toxicants, we're maybe a little more agnostic. But of course, we know that the drugs are pretty promiscuous too, right? I'm thinking of the recent story about ibuprofen causing male infertility. That wasn't one of its intended effects. So I guess the question is, given that this technology is so accelerating in terms of discovery of pathways and impacts and everything, and I am assuming that the drug companies are not just applying these technologies in the discovery phase, but also in the safety phases and the regulatory approval phases. Are we seeing a lot of cross-communication between we saw this 10, 15 years ago with the transcriptomics, a lot of crosstalk between the pharmaceutical and the environmental toxicology communities. Are we seeing that in terms of the toxicity side, this kind of crosstalk between the industry and academic researchers? And if not, shouldn't we be seeing more? Yeah. And maybe that's just a comment and doesn't.
Yeah, I don't know the answer to that question, but I was struck by the fact that there we haven't heard any pharmaceutical company perspective at this meeting. And I think, they are, I mean, aside from drug development, they're very concerned with adverse events. And so that's something where toxicologists and pharmaceutical companies, or toxicologists at pharmaceutical companies, would have a stake in how we apply these technologies. So I think that's something, if anyone in the audience knows what kind of dialogue has occurred already, I'd be interested in hearing that.
So I can say we invited several people from pharmaceutical industries to attend, but because they're developing these tools for proprietary use, they weren't prepared to share it at this workshop.
Linda Wennerberg from NASA. As one of the people who is going to be talking about decision making, the issue is with the talks were fabulous and very helpful. I'm seeing a lot about how screening can be done. It seems screening still, frankly, in its early stages, where you're looking at one by one things. We know that, and the question I would ask of everybody is, when we frankly do a lot of our work in terms of making decisions on regulatory levels and so on, one by one with one study. And so the question is to everyone is, knowing what the kinds of the tools are rapidly evolving, what is the effort to get something really, which is trying to actually really capture more of an actual system versus simply a one-off. And I'm not disrespecting the importance of doing this in steps to learn how to do one thing and then the other. I'm just asking, that's really where we're going to get real answers with these.
Yeah, I think that question is similar to the previous question about mixtures. And listening to your talk, Keith, I was kind of wondering to what extent the ToxCast and Tox21 programs have started to do mixture studies and start looking at transcriptional profiles and pathway activation in the presence of combinatorial chemical exposures. And it may be that there's still so much to be done with the single chemical approach that we're not there yet.
I mean, I can give you, I've personally been very interested in mixtures the entire time I've been at the agency, and we've just never gotten the momentum to get something going over that. I think we already have so many things on our plate that, and the mixtures really is it's just a huge combinatorial problem. I'm not really sure, I mean, off the top of my head, I hadn't thought about it in connection specifically with genome editing tools, whether that would give us any major advantages in looking at mixtures or not. But certainly mixtures are important. I think if there are biological models, like I said, that we could design bioassays for that involved modulating various targets that may be synergistic, that the genome editing tools would be a really useful way to do that.
I've been interested in that for some time because a lot of our patients smoke, and then they also will get exposed to some type of alkylating agent. And so I've been curious to know, what does that mean? And we know that smoking definitely decreases your ovarian reserve and can have lasting transgenerational effects for not only the F1 but F2 generations. And so at least that's been shown in mice. And so I would really like to maybe talk after about how we could try to group different exposures together and common exposures, and for me, exposures that are gonatotoxic. So.
And just to comment, one of the things that these talks were very good about was also bringing up the fact of not only things that are deleterious, but also beneficial, and trying to get a real sense of them. We chase that which is deleterious, and we should, but we also, a part of that understanding is underlying that there may be a series of protective measures or other measures that are extremely important. And in the way we traditionally do decision making, we're not thinking of those things. We're not aware of those things. We're simply looking at the deleterious action, that one deleterious action.
Lopin Zhang from UC Berkeley. I just have a comment for Dr. Rebecca Frye's talk. I really enjoy your talk and also your thoughts to put how to make the CRISPR screening in the cell system and to apply it to the human. So So I think that identify susceptible genes to identify susceptible populations is really important step. But also, I think last night at the dinner table, a few of us we will discuss is for CRISPR, you know, you are not only identify a single sort of gene to trying to test in human, but maybe, you know, going through the pathway analysis, and you actually identify a group of genes. And so that's how can help also us to identify the pathways and the mechanisms, so which could also help us to test that specific biological rules or MOAs in the human population. But I like to read, because when I was trying to put my talk together, I really had a trouble to see what else we can learn from the CRISPR screening, either in the cell system or in the animal model like Rick was presented, to really get into our ultimate goal is understanding the human population. So I just want to make a comment and also see if we can even consider about that or can brainstorm that. Another question is, how could we learn from or using the data we obtained from the CRISPR genomic screening? So I mentioned the pathway analysis, and I heard a cases talk. You had some thoughts about pathway analysis. So far, most of the people still based on the cytoscape, the Kiwi pathway analysis. So, I want to hear from you. I thought that you think that's not enough. What we should develop the pathway analysis. I think the pathway analysis would help us to really understand better or more from the data we obtained from at least the genomic-wide screening, CRISPR screening.
I don't have a good answer for you. I just, right now, I think it's a really complex problem because there's a lot of variables, and nature just has so much redundancy, and so many of the genes, the proteins are used in so many different pathways. It becomes statistically a really big challenge. And my opinion in the way that it's used a lot of times right now is you do a gene expression analysis and you get a list of modulated pathways, and you get 10 or 20 top pathways, and you pick 3 or 4 of them that fit your hypothesis, and you say, yes, my hypothesis was proven true. But it's not a very rigorous way to analyze that. But I don't know. I think we need to understand more about biology, and there's so much going on now at the big level, the systems type approaches, as that information becomes more analyzed and disseminated, and I think it'll eventually impact the way we look at pathways, and I think there'll be new statistical methods of doing that analysis, but I don't have anything specific I can add to that.
This is just a comment. I've been struck over the past 2 days listening to all the presentations by the very different approaches that people are taking to trying to understand chemical effects. And at the end of the day, I think it's going to be a combination of all these complementary approaches that's going to lead to understanding. So the high throughput bioassays and transcriptomics analysis, the genome-wide CRISPR screens, and the focused target validation and mechanistic hypothesis testing research is all going to complement each other to give us increased understanding. And I think I've been really excited by sort of hearing all these different approaches presented in one place and addressing a common problem. And CRISPR will be part of that, but it's not the whole. CRISPR is not going to replace all the stuff that you do in ToxCast and Tox21. It's going to enhance it, and so I think it's very exciting.
So I would echo your point, Mark, and I guess one last thing, because I know we're almost done here, as I think about using new technologies for risk assessment, policy decisions. I know one of the things that always comes up is really having confidence in the methodologies, which then leads you to have confidence about the research findings. And Mark, I was kind of struck by one of the things you pointed out in your talk yesterday was the difference between the H receptor knockout versus the knockdown, and that it appeared that in the knockout there were some potentially compensatory mechanisms coming up. I know, Rick, you mentioned something the other day, yesterday, that, you know, although we're using these gene editing tools to make specific changes, but there may be additional things that are happening that we may not be aware of, other mutations or other edits. How do we address those? Because that all gets back to the confidence in the findings that we get from the models we set up using these tools. And is that going to be a major hurdle still for us in terms of using gene editing for risk assessment, policy decisions, and for that matter, even medical-based decisions? I don't know if anybody wants to.
Well, as I said yesterday, I think the possibility of various mechanisms of compensation after complete knockouts, it's going to lead to some false negatives. And I talked to Chris about this yesterday in terms of the genome-wide CRISPR screens. What's not being picked up because the cells are compensating for the loss of function, and how do we understand that? So I don't know what the answer to your question is, but I think it is an issue that we have to keep in mind, and ultimately it's going to inform us about how cells work if we understand some of these compensatory mechanisms. It's a question in biology, not a question in environmental health sciences specifically, but it's something we're going to learn from the people who are really focusing on trying to understand some of these processes.
Yeah, I just want to add, you know, we have a large body of historical toxicology data that we know a lot about a lot of chemicals, how they behave, and what we've mainly been talking about are building models of various sorts, and we can use what we already know about those chemicals to validate those models. If they if the problems such as you were talking about yesterday are there and they're significant, then we should see that when chemicals don't behave as we expect them to. So I can't overemphasize the reliance on what I call reference chemicals, the gold standards. Your model needs to reflect the behavior that it should.
David, it's kind of the old adage, embrace the unexpected result, because it may be telling you something important.
So I just want to make sure we don't lose the thread of one of your sub-questions, which was, how do we make sure that the phenotype that we observed was caused by the mutation that we generated using the CRISPR? And I'm always a little bit puzzled that so many people are able to publish papers where they derive one cell line where they knock out a gene, for example, and they observe a phenotype, and that's the end of the story. I always want to see, even as a reviewer, I want to see them use 2 different guide RNAs and make even 2 cell lines where they knock out the same gene and get the same phenotype, because that would pretty well exclude the idea that there's a secondary mutation that accounts for that. And that's particularly the case when you're working with a cancer-derived cell line, because I think all of us who do cell biology know that they're shuffling their genomes all the time.
Yeah. And Rebecca, I think you were going to follow up on that.
I wasn't going to respond necessarily to that one directly, but I think, certainly we need guidelines, we need guidance, we need quality control. We probably need a set of minimum standards or something for using these tools in any field, environmental health science being one. But one of the things that I'm struck by is I really would like to challenge us to move beyond using these tools for understanding mechanism to intervention and solutions. So how do we modify someone's vulnerability to an environmental toxicant? How do we change them from being vulnerable or susceptible? How do we provide resistance, right? That would be the dream, right? If we can understand, of course we have to understand the mechanism first, but I think what would be success 5 or 10 years from now of these new tools is to say, here's an organism that is extremely sensitive or vulnerable to an environmental insult. Can we use these tools to protect that organism somehow? And of course, we have to understand first how that happens, whether it's a gene or pathway. So anyway, just as a statement, I think we may want to be pushing the boundary a little bit more than just trying to identify the genes and pathways to, right, to take that yellow mouse and make it brown again, right?
Okay, thank you very much, each of you, for participating in the panel. And I think we'll take a very short break because we're a little bit over time, and we'll return at 1:15. Thank you.